All right. Good morning, everyone. I would like to, I don't know what to either apologize because last week I actually came and when I am about to enter the class, I discover that I received a WhatsApp message that the class is holding with another person. So until then, I realized that you guys are occupied. So I have to go down and even confirm whether you guys did engage or the person couldn't show off. But unfortunately, I learned that you guys are doing some presentation. And unfortunately, today I am here and none of you is there. And the class is even locked. So I felt with two classes because last week we were supposed to discuss nutritional disorders, or nutritional anemia rather. This week, based on what we plan to do, this week we're supposed to discuss on iodine deficiency and fluoride deficiency. But unfortunately, none of these classes hold. So I decide instead of, because we don't have much time, instead of waiting to next week, I decide to record myself and share with you so that, remember, I asked you to read on this. So whatever you read, after following through my tutorial lecture, then you can compare. If there is anything you need to ask, maybe in the next class, we can meet. And I wanted to even see if I can make it, I can produce another tutorial recorded class where I will cover iodine deficiency and fluoride deficiency, so that by next week, when I enter the class, then we can see if there is any question regarding the recorded lectures, then we can attend to the questions. Without that, then we can proceed with our last meeting, which will basically be on immunity, infection, and nutrition. All right, so without taking much of our time, we're going to discuss nutritional anemia. And as a matter of fact, one of the most important things we need to know here is what is anemia right from beginning. Knowing anemia, and then what are the symptoms of anemia, because we are not going to talk about anemia with regard to symptoms of anemia with regard to other conditions like either iron deficiency, folic acid deficiency, or vitamin B12 deficiency, because the symptoms are basically the same. In a few cases, or in a few cases where we have exception, then we're going to talk about the exception. But basically, anemia is characterized as the condition with a low hemoglobin or red blood cell count. So when we have a low hemoglobin concentration or low red blood cell count, that condition is regarded as anemia. And there are quite a number of reasons that may give rise to anemic condition, a lot of pathological condition, because on itself, anemia can also be considered as a symptom rather than the disease condition itself. So why or how is the decrease in the red blood cell count, would a decrease in the concentration of hemoglobin affect the system? What we know about red blood cells, the basic function is that they are involved in the transport of oxygen, and transporting oxygen from lungs to different parts of the body where cellular respiration takes place. And we know the role of oxygen in cellular respiration, where we mentioned, maybe for those of you that did some courses on that, cellular energy metabolism, they must have discussed about electron transport chain, where we mentioned that oxygen is the final electron acceptor in electron transport chain. So oxygen is actually involved in the transfer of electron transport chain during oxidative phosphorylation or energy production. So that process led to the production of energy in the system, the overall energy production in the system. So from there, we can even extrapolate some of the symptoms associated with this anemia, because when you have a decrease in a red blood cell or hemoglobin count, then definitely you will have low oxygen concentration, and then low oxygen concentration will affect energy production, and hence you will have low energy production in the system. And symptoms of low energy production in the systems are too clear. You can talk about weakness of the body. You can talk about dizziness. You can talk about somebody being tired easily. You can talk about fatigue, etc. And another thing also, difficulty breathing, because the inadequate oxygen that are needed in the system can bring about difficulty in breathing, and that can also relate to the increase in irregular heartbeats, because the amount of blood is not enough, so the heart will be beating irregular. And in addition to that, also we have pale skin. The skin can, from these two images, you can see the two hands. One is looking pale, the other one is looking reddish, which simply indicate the availability or the presence of red blood cells available. Some other symptoms like cognitive impairment among the children, headache, etc., are all related to anemia. So we're going to see the causes of this anemia generally, even though whenever we are discussing the individual anemia, we are then going to see an individual causes in each cases. But generally, we have malnutrition, which has to do with deficiency of some particular nutrients. And we have chronic infection. Some chronic infection might be a situation like malaria infection, where the red blood cells that are affected. In that situation, you can have, because in the cases of malaria, it involves destroying of red blood cells, and so quite a number of other diseases that also directly involve destroying the red blood cells. So that can also lead to anemia. And in addition, other infection could also indirectly relate to anemia, because you can have some organisms that can actually make use of some nutrient, just like we mentioned the nutrient. So they can use those nutrients available, and as such, those nutrients are related to either red blood cells production or hemoglobin production, as in the case of what we are going to shortly discuss. We also have inherited these disorders. We can have disorders that affect some certain genes that are related to this red blood cells production, erythropoiesis, or rather in the synthesis of hemoglobin. We also see some medications, some drugs taken that can actually affect some of the reactions that are involved in either cell formation generally, which the red blood cells are one of the major targets, because when we're talking about cell formation or cell division, red blood cells, or let's say the erythropoiesis, is one of the processes that are involved in the synthesis of red blood cells, and targeting synthesis of cell, red blood cells are one of the major targets, because generally, hematopoiesis takes place in the bone marrow, and they are one of the cells that are rapidly produced in the system because they have a lifespan. In addition to other cells like embryonic cells, there are cells that are being produced rapidly in the system. So if any drug is taken that can inhibit synthesis of a cell, because I'm talking about synthesis of a cell. Specifically, you will see a particular reaction or a particular pathway that is very important. Let me say that is very necessary for cell division to take place. So you can have some drugs targeting this particular reactions, most especially drugs that has been used in the cancer treatment, and some drugs even used in the treatment of infection, targeting some particular bacteria. So those drugs can actually act and then inhibit the synthesis of most rapidly growing cells. And then there are cases of autoimmune disorders where the system will produce some proteins that can attack some certain cells or receptors in the system, thereby impeding their own activity. And then we talk about blood loss, how an individuals can either it's an orphan, or an internal or external blood loss. In the case of hemorrhage, there are quite a number of disease conditions that can cause internal hemorrhage, and even the loss of blood, physical or external loss of blood, also lead to anemia. So before then, what is this nutritional anemia? Given the definition of anemia, the nutritional anemia, we can say specifically is an anemia or low red blood cell counts or low hemoglobin concentration due to a nutrient deficiency. And based on this, they are classified into three, based on the size of the red blood cells and the liking nutrient, because each of the liking nutrients have a specific symptoms that are related. When you read more of hematophosis and the stages of hematophosis, you will get to understand why micro and why macro and microcytic, because it has to do with the formation during the process of the cell division. Just before cell division, you can have a cell division that containing red blood cells that are bigger than the normal size, or red blood cells that are smaller than the normal size, which has to do with the issue related to the division, dikinesis, or rather cytokinesis, where you have an inadequate separations of the genomic materials. So we have nomocytic, microcytic, and macrocytic. So on the basis of this course, nomocytic is when you have the number of red blood cells count to be low, but the red blood cells maintain their normal size, which mostly are the cases you will find out in protein energy malnutrition, where the requirement for the synthesis of red blood cells are lacking generally. So you can have the red blood cells, but they're very low in number, but their size are normal. But in the case of microcytic, is when you have the red blood cells being smaller than their normal size. And basically, this happens when you have deficiency of iron, copper, and vitamin B6, which is pyridoxine. And you also have microcytic when you have the red blood cells bigger than their usual size. And this occur usually when you have deficiency in either folic acid or vitamin B12. So for the sake of this class, we are going to cover the most common, most prevalent cases, which are iron deficiency, folic acid deficiency, and B12 deficiency. Other deficiencies leading to nutritional anemia, apart from the copper and the pyridoxine, include selenium also. That maybe when you read, you can also come across quite a number of similar cases that lead to anemia. Okay. Now, let's look into iron deficiency anemia. Apart from the function of iron in boosting immune system as well as the maintenance of brain function, we also know that iron is involved in the synthesis of hemoglobin, and later on we talk about already how hemoglobin is involved in the transport of oxygen, because the hemoglobin in the red blood cells is involved, is the main a binding target or binding point of oxygen, where oxygen binds to and then transport to different part of the body. And iron deficiency, one thing we need to know, is the most prevalent nutritional disorder, mostly in infant and middle-aged women. And the daily requirement of this iron is about, I'm talking about the recommended daily allowance, is recommended dietary intake, sorry, is about 1 milligram, and male can store up to about 1 gram, while females only about 300 milligram. And this has to do with the loss of blood related to menstruation and the rest. That is why, because we're going to see how iron is being stored in the body, and how is this iron loss related or are most common in women than in men, and why men were able to store this more than what our women stored. And then we talk about these ions exist in two major forms. Basically, we iron that are found mostly in animal source. The animal source of iron basically come in form of heme iron. This type of iron usually doesn't have a problem after ingestion. They can easily absorb by the body. But the non-heme iron, which are mostly found in plants, has to undergo some processing, oxidation reduction reaction before it gets absorbed into the system, and then the body will make use of it. So we have quite a number of sources of iron here between the vegetables, red meats, fish, egg, seeds, cereals, etc. They all contain high amounts of iron. So we talk about the pathophysiology. How does the body take care of iron? So when you take a food that contain iron, usually this iron come in form of iron 2 bound to a compound, or a compound attached to iron. So iron usually exists in ferrous iron, iron 2. But when you take in a food containing iron, and then this iron bound to some organic molecules, once they reach the stomach, because of the acidic nature of the stomach, this will, their digestion and whatever will causes the release of iron. So once the iron is released, it is released as iron 3. And iron 3 normally cannot get absorbed easily into the system. So the iron 3 will be converted to ferrous iron, which is iron 2. So iron 2 basically will be absorbed with the help of a carrier protein called heme carrier protein. So heme carrier protein export or facilitate the movement of iron through divalent metal transporter. The divalent metal transporter will transport iron, and once the iron reach the enterocytes in form of iron 2, then another enzyme can oxidize the iron to form iron 3, which can be stored in form of ferritin. While if the iron is needed in the organs, in other organ of the body, the iron can directly move through another pluripotent transporter, which is an iron-regulated gene 1 transporter that can be transported to into the cellular circulation, into the plasma circulation, or rather blood circulation. in the blood circulation with the help of other protein like ceruloplasmin or peroxidase. 2, iron 2 can be converted to iron 3, and iron 3 will bind to transferrin, and transferrin is nothing but a transporter protein which can transport iron to different parts of the liver. Iron can be converted to ferritin, which is the storage. You can see iron can be stored in the erythrocytes, which are the mucosal cells of the GIT, or can also be stored in the liver in form of ferritin or hemosiderin. So also this iron can be taken to bone marrow where hemoglobin are synthesized for red blood cell synthesis, and can also be taken to muscles where myoglobin, just like hemoglobin in the muscle, in the red blood cells, the myoglobin are also synthesized in the muscles, and other tissues for the synthesis of cytochromes and then non-Hems iron. So apart from the storage here, we then talk about the majority or the vast amount of this iron after taken, because you can see the body control this absorption of iron using a particular hormone called hepcidin. So at least in a day, even when you take more than the requirement, just about 1 to 2 of that milligram needed for the body will be taken. And once it is taken, about 75% of what was taken will definitely be used for hemoglobin synthesis, while only 10 to 20% are being stored in the form of ferritin, while the remaining are used for other synthesis of other molecules, from non-Hem ions to the cytochrome synthesis. And every day, due to the disquamation of epithelial cells, but even though in that cases, you would have a few distance, like the process of apoptosis, programmed cell death of the epithelial cells. Some of these iron get lost. But apart from this process, this is the only non-process where iron get lost out of the body, unless when there is bleeding, internal or external, is one of the major processes where iron, but there is no physiological process where iron are excreted out of the body. So you can see out of what was taken into the body, only about 10 to 20% is actually being stored in form. So the storage is not as big or huge as compared to what was used in the case of hemoglobin synthesis. So in hemoglobin synthesis, in the process of blood loss when it comes to menstruation, so what is lost is the high amount of the blood of the iron in the body is being lost during menstruation. That is why the fluctuation in terms of iron content in females than in males. So we then talk about the causes of iron deficiency. What causes deficiency in iron? We say inadequate intake. So maybe you're taking a food that doesn't contain iron, which is mostly the very rare cases, mostly. But other like transports or storage, you will have a problem with transportation of iron, and have a problem with the storage of iron mixed in those transporter molecules. We mentioned the role of divalent metal transporter. We also talk about the role of periprotein, which are the transporter molecules that facilitate the movement of the ferrous ion into the enterocytes as well as to the plasma circulation. So when we have a defect in one of these molecules that are involved in the transportation, we also talk about transferrin that are involved in transporting in the plasma fluid, which is facilitate the movement to different parts of the body. So We also have problem with absorption. We have a problem of chronic blood loss, which I just mentioned. We also talked about infection, hookworm infection, or any type of infection that can make the iron not available for hemoglobin synthesis. And these are the basic reasons that may lead to iron deficiency anemia. So we talk about treatment. Treatment basically started with fortification, but that is when the situation is not worse, then followed by supplementation of iron in form of iron supplements that can be taken or prescribed by a medical personnel. And if the situation is a bit worse, then you go to oral iron therapy, where usually in form of perosulfate or gluconate fumarate, and iron can be given to an individual. Or when the situation is worse, also, like in severe cases, you can have IV or IM, where an iron can be ingested into the body directly. But in most cases, this is a situation when the absorption, the problem related to absorption. So even when you were given a supplement, you will have a problem. Even when you were given an oral iron therapy, be a problem. So the next option is to go for an IV or IM, that the iron can directly move into the blood circulation and then take into all the part of the body where it is needed. Okay, before then, I think I need to go back. We talk about laboratory assessment. What are those parameters that are needed in diagnosis of iron deficiency? I think so. Basically, in the case of iron deficiency, like the serum level of ferritin, the level of transferrin, serum level of transferrin, and serum level of protoporphyrin. Protoporphyrin is a compound that is synthesized or produced during the synthesis of hemoglobin. For those of you that have an experience on biochemical reactions involved in the synthesis of porphyrins, and we know porphyrin ring, the main heme ring of hemoglobin. So the analysis of protoporphyrin will tell an individual whether the level of iron in the body is lacking or not. So either ferritin, transferrin, or protoporphyrin level can be assessed to give an indication of iron deficiency in the system. All right. So I decided to segment the classes so that in case of network failure, so we are done with iron deficiency. If you have any question on iron deficiency, then in our class, then we can rest and then we can attempt to those questions. Now let's talk about folic acid deficiency. Folic acid deficiency, folic acid is mostly found in leafy vegetables. And one thing we know about folic acid is that it played a very important role in carbon metabolism. In carbon metabolism, we can remember quite a number of reactions where folic acid involved in transfer of one carbon or addition of one carbon, basically carbonation, let's say carbonation, but depending on, because you can have this transfer of carbon in different oxidation state. So we have this folic acid activated to form tetrahydrofolate once it is taken in the system. But basically, in the diet, it exists as what we call polyglutamate. So once it is taken into the system, it is then converted to monoglutamate by an enzyme folate conjugase. So the daily requirement of folic acid in adults is mostly 400 microgram, and then the body can store up to 10 to 12 milligram in the liver. So naturally, you can have folic acid in a different type of food. We mentioned leafy vegetables, which must still stored in abundant. And then we have, because the body store it in the liver, so when you take liver, you can have abundant of it. So apart from that, we have quite a number of juice like orange juice, pineapple, tomato, in some fruits, then and also in some insect, you can have a lot of folic acid present. So we talk about causes of folic acid deficiency, even though I think I need to talk about pathophysiology, but nevertheless, we can still talk about the dietary intake. Dietary intake, when you take a food that are deficient in folic acid, it can lead to that. But what causes that in most cases are inability to take, inability to absorb, apart from the fact that you can take a food that does not contain this, you can also take a food that contain this folic acid, but the body is unable to absorb it. This in the case of malabsorption. So there are quite a number of cases that lead to that. You can have a problem of old age, aging. We know aging comes with a lot of challenges, and the process of aging, which basically has to do with, I think, if you can remember, one of the theory of aging, apart from the most common one involving telomerase, and then we have what they call this radical theory of aging that has to do with the generation of reactive oxygen species over time. This wear and tear theory that has to do with the overuse of something over a long period of time can reduce efficiency. So also the same thing happened in the case of cells. When the cells matured for a longer period of time, being used and reused, at a point they become tired, and inefficiency in function may set in, and then you can have those receptors involved in the transfer of folic acid into the systems being not efficient, and you can have deficiency in the case of alcoholism, in the case of chronic disease, which I earlier explained. You can also, which this is the same thing with the case of malabsorption, and you have also a situation where you have an increase in the requirement of this folic acid, like during pregnancy, when an individual is no longer alone. Apart from your own requirement, you have the requirement of the growing fetus. And the growing fetus also is an embryo, and the cells in embryo are one of the most rapidly growing cells, rapidly multiplying cells, and of course they need a lot of this folic acid. And infancy also, the growth in that infancy is also high, so the cells divide faster at infancy, so the need for this folic acid is also high at that situation. Like malignancy also, when you have a problem like cancerous situation, where you have a growth in the cells, that the cells grow faster, and that situation also you can have high requirement of this. Also, some drugs, just like we mentioned, where you have some certain drugs that are involved in inhibition of some certain, like in cancer cells, we have drugs that inhibit the hydrofolate reductase. Also, we're going to talk about see the reaction involved in the hydrofolate reductase, and this process generally leads to inhibition in the utilization of folic acid in the systems. And once folic acid is inhibited, we're going to see a particular reaction that is centrally with regard to synthesis of genetic material, because the synthesis of genetic material involves the formation of this nucleotide bases for the synthesis of genetic material. And when you look at the cell cycle, the post-stage in cell cycle, you have to first of all synthesize all genetic material so that you can have a duplicate cell. So before you do that, you synthesize the genetic material. For you to synthesize genetic material, you have all the necessary requirements for the synthesis of the genetic material. So what are the requirements in the synthesis of a DNA? Are the nucleotides. So those nucleotides, you have to synthesize them. So we can see how nucleotides are synthesized. First of all, before you synthesize thiamine nucleotide, first the uridine nucleotide is synthesized, and the uridine nucleotide has to be converted to thiamine nucleotides, and converting thiamine nucleotide now to uridine nucleotide. Sorry, converting uridine nucleotide to thiamine nucleotide is a problem that or is a situation that involves the use of folic acid. So that particular reaction can be affected, and as such, the cells that is growing and could not have one of the basic requirements in DNA synthesis, then there will be a problem in that case. So there are quite a number of cases. We also talk about oral contraceptives. So oral contraceptives, the oral contraceptives, majorly some of them inhibit or serve as an inhibitor of folate conjugates. And we mentioned the role of folate conjugates in converting the polyglutamate to monoglutamate, so that it can easily be absorbed by the system. So here we can have the folic acid, if taken as a supplement as folic acid, it can go directly absorbed by the system. But if it comes in form of polyglutamate, that enzyme will have to convert the polyglutamate to monoglutamate, and then it is absorbed by the system. Or in form of supplement, usually comes as 5-methyl tetrahydrofolate. So once it enters the intestinal mucosa, the folic acid can be converted to dihydrofolate. Dihydrofolate will be converted to tetrahydrofolate by dihydrofolate reductase. So this is the inhibitor I talked about. So you have this expression of this enzyme dihydrofolate in the enterocyte. You also have it in the inside the cells. So any drug that can inhibit this enzyme can actually stop or inhibit the utilization of folic acid. So we have dihydrofolate reductase to form dihydrofolate. We also have dihydrofolate reductase to convert further to tetrahydrofolate. Tetrahydrofolate now can be converted to 5-10 methylene tetrahydrofolate, which is then also further converted to 5-methyl tetrahydrofolate. So in either case, in the process of one carbon transfer, either methylene tetrahydrofolate, 5-methyl tetrahydrofolate is involved, 4-methyl tetrahydrofolates are involved, etc., etc. Quite a number of them are involved. So we'll see how this 5-methyl tetrahydrofolates can be used in the synthesis of. Okay, now after taking, you have this converted to dihydrofolate by dihydrofolate reductase, tetrahydrofolate to methylene tetrahydrofolate to methyl tetrahydrofolate, and methyl tetrahydrofolate is involved in the synthesis of, for example, in the previous slide, we can see 5-methyl tetrahydrofolate can go directly into the system. And even though 5,10-methylene is produced, but basically the body absorbs folic acid in form of 5-methyl tetrahydrofolate. Only a few concentration of it is being absorbed in form of folic acid. So 5-methyl tetrahydrofolate is what is majorly available in the cells. So you can see this methyl tetrahydrofolate, before it can be converted to methylene tetrahydrofolate, and the methylene tetrahydrofolate is involved now in this conversion of the oxyuridine monophosphate to the oxythiamine monophosphate. It has to be first of all converted to tetrahydrofolate. Then tetrahydrofolate by tetrahydrofolate reductase, dihydrofolate reductase, then it converted to methylene tetrahydrofolate, reductase. So what is important here is vitamin B12 has to be there. So vitamin B12, in the presence of vitamin B12, methylene tetrahydrofolate is converted to tetrahydrofolate, and vitamin B12 becomes methyl B12 or methyl, which is vitamin B12 is cobalamin, which will be methyl cobalamin. We'll talk about this role of methylcobalamin in the conversion of homocysteine to methionine in the case of vitamin B12 deficiency, and how vitamin B12 deficiency is related to folic acid deficiency. But this is basically the connection. We'll talk about that when we come to vitamin B12 deficiency. So you can see the tetrahydrofolate is formed here. And once the tetrahydrofolate is formed, it's converted to methylene tetrahydrofolate, and then methylene tetrahydrofolate will now be used for the conversion of this deoxyuridine monophosphate to deoxythiamine monophosphate, which finally converted to deoxythiamine triphosphate used in the synthesis of DNA. And once this pathway been inhibited, then there are going to be a synthesis of DNA, there are going to be a cell division, etc. Now, how is the deficiency can be treated? Just like the explanation we gave, it can be food can be fortified with folic acid fortification, which I think you nutrition student you will come across a lot of food that were fortified with folic acid depending on certain conditions targeted. You also have a folic acid supplementation, which you all know. We also have oral therapy, and the last one, which is folic acid injection in the cases of the problem involving malabsorption, where the folic acid cannot be absorbed by the system, impairment of the absorption, maybe autoimmune disorders that are related or affecting the absorption process. Then folic acid needs to be injected directly, either in form of intravenous or intramuscular, so that it can easily be utilized. So the problem of storage itself, if the folic acid cannot be stored, therefore somebody can continuously be receiving folic acid injection so that it can easily be used by the system. So this is all about folic acid deficiency. And then we talk about vitamin B12 deficiency. In vitamin B12 deficiency, one thing we need to know is that vitamin B12 is produced by microorganisms, like the case of folic acid, which is majorly the vegetables produce is folic acid. So vitamin B12 is produced by microorganisms, and from the little distance we've seen that vitamin B12 is involved also in carbon metabolism, and it is the mostly seen in elderly individuals because of malabsorption problems. So deficiency of cobalamin has to do with mostly malabsorption in elderly people, individuals, because the storage form of it, the body doesn't need much of it, and the body is stored a lot of this. So when you have a problem of malabsorption, that is when the storage will be depleted to the point that it's remain nothing. And then the next thing is the treatment and how does the body take care of it. So even when you take a food containing in form of supplement, oral, whatever, in form of fortified food, you will never have your problems solved. Rather, you may have to go for maybe intravenous injection, because the issue has to do with malabsorption. So we have two major active forms, which include methylcobalamin and 5-deoxyadenosine cobalamin. So one of the difference we mentioned with regard to symptoms of anemia, we cite the symptoms are the same, only in some few cases. So here we are going to talk about some cases that are specific to cobalamin deficiency, which is neurological disorder. We'll see the neurological manifestation. that's related to cobalamin deficiency, which is different in other cases. So sources of, we said microorganisms, basically anaerobic microorganisms, and animals basically absorb or obtain this, either the ones synthesized by intestinal bacteria or the ones that is attached to food in the form of liver, kidney, milk, egg, etc., etc. The plant cannot actually synthesize vitamin B12, so you will not find it in plant source. So what are the causes of this? I've already said basically has to do with malabsorption. So you can have an autoimmune destruction of gastric parietal cells. We'll see what are these gastric parietal cells and how they are involved in absorption of vitamin B12. You can have also a hereditary problem, maybe you inherited a defective gene that are involved in the absorption process, or you can have a kind of surgery that affects these proteins that are involved in the digestion process, or you can have an insufficient production of these molecules also. Basically, we call them intrinsic factors that are involved in the facilitation of the absorption of this. You can also have the deficiency in individuals that are strict vegetarians. So when you are a strict vegetarian, you don't take any animal source, then you have a high risk of developing cobalamin deficiency. So what happened actually, what is this intrinsic factor? Because while discussing the causes, you see, we talk about autoimmune destruction of gastric parietal cells, we talk about hereditary malabsorption, we talk about partial or total gastrotomy, insufficient production of intrinsic factor. So all these are all tied in on intrinsic factor. There are a lot of intrinsic factor. And then the last one is only in the case of vegetarian, where they don't actually take these things. They only take plant source. So plant source, and as we said, they don't produce cobalamin. So we have, when you take a food containing this vitamin B, vitamin B12 is actually found attached to protein. So the gastric acidic nature of the stomach will now cause the hydrolysis, thereby separating the protein with vitamin B12, just like we explained in the case of iron. So we have this vitamin B12 release. So vitamin B12 will now interact with the intrinsic factor that is produced by gastric parietal cells in the stomach. There is reason when we say when you have a problem with the gastric parietal cells, they may fail to produce this intrinsic factor. So usually there are some autoimmune disease that can actually affect gastric parietal cells, make them unavailable, will make them unable to produce in these intrinsic factors. So or you will have the intrinsic factors that have been produced not enough, or you will have the intrinsic factors that are defective, that we said hereditary issue, that are very defective, so they are unable to perform their function. So when this intrinsic factor binds to vitamin B12, they then absorbed, facilitate the absorption into the mucosal cells. And the mucosal cells, they become methylated to methylcobalamin, and then methylcobalamin is then absorbed into the plasma. In the plasma, we have transcobalamin that is involved in the movement of this cobalamin to different part of the body, to the tissue, where they can be stored, where they converted to in the liver, where they are converted to oxyadenosyl cobalamin, which is the storage form of cobalamin. So we'll see the pathopathology. How is this involved in one carbon metabolism? We have two examples, and then the last one, one is how it is related to folic acid, leading to what we call functional folate deficiency. So we can remember fatty acid oxidation in the case of odd chain fatty acids, where you have, because the fatty acid oxidation actually remove continuously two carbon atoms. So in the case of odd chain, the last carbon atoms you will have will be 3, which leads to formation of what we call prophenyl-CoA. This prophenyl-CoA is converted to methyl malonyl-CoA. So the same thing also when you are talking about a breakdown of some amino acid like valine, isoleucine, threonine, and methionine, also thiamine and uracil, the case of nucleotide bases. So you produce methyl malonyl-CoA. So methyl malonyl-CoA can be converted by methyl malonyl-CoA mutase to succinyl-CoA. And then succinyl-CoA downstream can be used, in citric acid cycle as one of the citric acid cycle metabolite, or can be used in the production of porphyrins. And we know what porphyrins are. We've mentioned porphyrins while discussing about iron deficiency. So you have methyl malonyl-CoA converted to succinyl-CoA, and this reaction is more of adjustment of a carbon, of a methyl carbon, and this process involves this 5-deoxyadenosine cobalamin for this adjustment. So if this is deficient, so you will have accumulation of this methyl malonyl-CoA. So you have methyl malonic acid being excreted in the urine. So one of the process in diagnosing this condition is checking the urine, assessing for methyl malonic acid. So when you have high concentration of this, it's an indication of this. In addition, there are other like serum vitamin B12 also is an indication. And you can also have transcobalamin 2 that are involved in transportation of cobalamin in the blood circulation. Also, when the serum level is high, it also tells that there is a problem with this thing. Sorry, when you have high level of this transcobalamin, it simply says that because the system is done in such a way that there is a signal that more of this should be available. So the signal will come in such a way that transcobalamin will be produced so that it can transport a lot of cobalamin. And when you check and the serum transcobalamin level is high, it's simply an indication that cobalamin is deficient. So you can directly check also the level of the cobalamin in the system. And you also another point where cobalamin is also involved is methyl cobalamin in the conversion of homocysteine to methionine. And we know how homocysteine, or where homocysteine comes from in the still also in one carbon metabolism, methionine react with the ATP to produce S-adenosyl methionine, and we know S-adenosyl methionine is also involved in transfer of a methyl group methylation, basically. So once it methylates, it becomes S-adenosyl homocysteine, where the base is released, and then you have the homocysteine, and then the homocysteine is being converted back to methionine, and the cycle continue like this. And what is responsible is the methyl cobalamin is involved in the conversion of homocysteine to methionine with the help of homocysteine methyl transferase. So this methyl cobalamin is also produced by methyl tetrahydrofolate. Remember, methyl tetrahydrofolate is basically like the major source of tetrahydrofolate to the system. And although it is involved in quite a number of reactions, just like this one, and this also serves as a point where other form of tetrahydrofolate can be produced so that it can perform the one carbon metabolism. So apart from this N5-methyl tetrahydrofolate, even the N5-N10 methylene tetrahydrofolate, methyl tetrahydrofolate, 4-mil tetrahydrofolate, etc., you have to have tetrahydrofolate first before all these were produced, and then they are involved in quite a number of reaction in the system. So if you have deficiency in cobalamin, it simply says the N5-methyl tetrahydrofolate cannot be converted to tetrahydrofolate. As such, quite a number of reactions in the system will behave as if tetrahydrofolate is, even when N5-methyl tetrahydrofolate from the diet is there available. This is classical of what we call folate trapping or functional folate deficiency because of the deficiency of methylcobalamin. That is why sometimes you can have deficiency in cobalamin, vitamin B12, but it's appear as if it is folic acid deficiency. So it's very difficult to even differentiate because the cobalamin causes that deficiency in folic acid because of this particular reaction, which make the tetrahydrofolate unavailable, and as such, tetrahydrofolate that it's used in the precursor of the synthesis of other tetrahydrofolates that are involved in 1-carbon metabolism, just like N5, N10 that are involved in the reaction we mentioned, where deoxyuridine monophosphate is converted to deoxythymine monophosphate is actually inhibited. So this is basically the explanation, and mention how this thing can be assessed. And then I already talked about the treatment. I think what is left here is just to mention how this thing is related to neurological problem. One thing we know is that malonyl-CoA is involved in the synthesis of, malonyl-CoA is there is accumulation of methyl malonyl-CoA. This accumulation of methyl malonyl-CoA in a situation like this may tempt the system to make use of methyl malonyl-CoA, this biosynthesis of fatty acids. Remember, malonyl-CoA is involved in the synthesis of fatty acids. Situation like this, when you have accumulation of this, the body tempts to use methyl malonyl-CoA. And once the methyl malonyl-CoA is used in the synthesis of fatty acid, then that fatty acid will be produced as the branch fatty acid. And the branch fatty acid, when used in the synthesis of cell membrane, more especially in the formation of myelin sheaths of neuronal cells, it will lead to a number of disruption in function of these myelin sheaths. And this is characterized with a lot of neurological manifestations, including peripheral neuropathy. You have numbness, you have paresthesia, which involve the tingling formation, sensation, like needle itching, loss of vibrational sense, etc., including visual change related. And one also diagnostic parameter involved in this is the intrinsic factor antibodies actually be used, or gastric parietal cells antibodies can be used to detect if this is actually functioning well. So we already talked about the treatment, which I mentioned. This is a problem, then IV is the most easiest solution. Even though you can have oral replacement in some cases, but if it failed, oral replacement like cyanocobalamin can be used, and hydroxycobalamin can actually be used in the case of injection over some period of time. If it's continued, then it is a case that an irreversible damage which need a lifetime therapy if necessary. So if you have any question, please, when we come for the next class, we discuss, and then I think I will try also and produce a similar thing in the case of iodine and fluoride deficiency, so that when I come next, we'll just discuss nutrition, immunity, and infection, how nutrition affects immunity, and how is that also related to infection. But meanwhile, you wait for the next recorded class. so that make sure you listen, you study. If you have a questions, you then jot your questions. When we come to the class, then we discuss, and then we move to the next distance. See you.